This is a production of Cornell University.
So I'm really happy to be here. I have to say this is not an invited talk, but I wanted to do it, and I asked Gillian if I could, and so she said yes. So I'm really very delighted to be here. And what you might want to ask is there more than one Phytophthora infestans? And I'd say the answer is yes. My thesis in this seminar will be to convince you that there are dramatically different versions of this organism. And let's see. Another question that I think we should address is, does it matter if there is more than one version of this organism? As Alan alluded, it turns out that this idea is not popular. And maybe it's inconvenient, but I will illustrate toward the end of the seminar how unpopular this idea is. My organization is listed here on the screen. Now, Kathy Hodge told me that I had to introduce the pathogen and indicate why it's important. So Kathy, for you, I'm going to do that. You've seen some of these slides before. And then I'm going to talk about my lab's motivation for addressing this question. We'll look at some differences, and then we'll look at some more differences, and then we'll look at some of the implications. So first I want to start off by describing the asexual cycle of Phytophthora infestans, and this is, again, many of you know this already. This is a sporangium. If placed under cool conditions and in the presence of water, zoospores can be produced from this sporangium. Here's a schematic of one. It's hard to get a picture of a swimming zoospore because they're swimming. If it lands on a leaf, it can produce a lesion like this, and that's about a 2 or 3 day old lesion. Within a couple days, those lesions expand. Overnight, there could be massive sporulation from those lesions. If you look at them under dissecting scope, as some of you might do so in the field, this is what they look like. You can see the sporangia among the trichomes of potatoes. And this is a microscopic view of these sporangia. They are aerially dispersed, and so these sporangia can be flown in the air or splash dispersed, and they can survive in the atmosphere for some hours. What's, I think, really important is that this whole cycle can take about 5 or 6 days. A single sporangium can give rise to a huge number of other sporangia here. We've measured up to 300,000 sporangia from a single lesion. So the reproductive potential of this organism is really important. And as a result, it can turn a field of healthy looking potatoes like this in 7 or 8 days to something that looks like that, to something that looks like this in another 7 or 8 days. So those are real pictures of real epidemics that I took and observed. So this organism is really important. It also has a sexual cycle. There are 2 mating types, A1 and A2. And the oospores are the long-lived spores that survive. They can survive in soil for many years. It turns out in nature or in agriculture, there are only 2 places in the world now where the sexual cycle occurs. One is in central Mexico, and the other is in the Scandinavian countries, and perhaps other parts of Northwest Europe. And as Alan mentioned, we've known about this organism for a long time. I've worked on it all my career. My mother used to say, well, Bill, have you solved the problem yet? She asked that question time and again, and I said, well, no, Mom, not yet. Toward the, later on, she stopped asking that question. But I'm not alone. There are a lot of us who have not solved it yet. It first appeared in the United States in 1843, first appeared in Europe in 1845, and as Alan said, it caused the famine in Ireland from 1845 for the next, really quite a few years. I think it originated in central Mexico, in the highlands of central Mexico, probably came first to the US and then to Europe, and then from Europe, it went all the rest of the world. It went on infected seed tubers. Potatoes are planted from tubers. Those tubers can get infected, and it turns out that Northwest Europe is a great place to grow seed tubers, and they are transported all around the world, and that's how this organism gets around. It's remarkably effective at going worldwide. It is, as Alan also mentioned, it's still a problem. We estimate $6 billion a year annually in losses and control costs, and more than 3,000 articles annually on this disease. Many of them are in the refereed literature, but some are not. And that's because it causes a disease in agriculture, is the reason that it's important. I'm going to give 3 quick examples from my experience of why this is an important pathogen. This is a picture taken, actually, my mother-in-law sent this to me. She lives south of Utica. The picture taken in 1995 of the Herald American from Utica. It's a picture of Byron Chapman, who the year before had lost his entire crop to late blight. A new strain had come in. It had devastated these fields. And he sent me these data indicating the magnitude of the loss that he experienced. You'll notice he says he had $1 million in losses. He will not grow potatoes in 1995. His crops were destroyed in 1994. Not only did he not grow potatoes in 1995, but he declared bankruptcy. And he actually died a couple years after this photo was taken. Some of you might remember this one. This is 2009. There was a major pandemic of late blight on tomatoes up and down the East Coast. And it caused a lot of problems because previously, late blight on tomatoes had been not so problematic. But it got a lot of people upset in the Northeast in 2009. This is an email I got from one of those persons who said she was an organic grower. She had 23 different varieties. They're all gone. She had to leave home as her husband is pulling and bagging all 63 plants. They had 100% loss. She was planning to sell tomatoes for an additional source of income. And my third example concerns another lineage. This is Blue 13. It originated somewhere in Northwest Europe. It also has gone all over the world. It, in 2014, caused a terrible epidemic in West Bengal. The previous strains that had been there were not so severe. Blue 13 was very aggressive. It was resistant to a dominant fungicide, mefenoxam or Ridomil. It was highly virulent, and it infected both potatoes and tomatoes. So just 3 examples that late blight is still a real important problem. So what I want to describe next is my motivation for addressing this question. Will the real Phytophthora infestans make itself known? And this is a question that my lab has been sort of toying with for the last 20 years. We attempted to get it funded, but that was never successful. But I'll tell you why we started investigating it. And I'm going to look at a lot of this as Fry Lab failures. These are some field experiments that we did some years ago. So there's a failure in each one of these experiments, and I'm going to describe the failure in this one. What you can see at the end of the season is that there are some plants that are healthy, and there are some plants that are dead. And what this experiment, among other things, was attempting to do was to investigate the role of osmotin. Now, this paper appeared in PNAS in 1994. And it says that osmotin overexpression in potato delays development of disease symptoms in late blight. That's fantastic. Here's a transgenic approach. I contacted the authors. They sent me their transgenic potatoes. We planted those and compared them with the non-transgenic. And unfortunately, this is the result. The plants were not resistant. We could measure no resistance in those transgenics. So this is one of our first. You know, they saw the resistance in the lab. We could not see it in the field. A second example occurred somewhat later. And this result from a colleague in Europe who wrote me, and it was really interesting. She said, I've created resistance to late blight. I said, wow, that's really neat. She licensed the technology to a company. And then lo and behold, they showed up in the US because they wanted to sell transgenic resistant potatoes in the US. They couldn't do it in Europe because of GMO attitudes there. So we said, yeah, great, let's do it. And so this is a picture of that experiment. There are about 100 different transgenic events or transformative events in this, this experiment with lots and lots and lots of replication. I think the technology was that there was a pathogen-specific promoter linked to an R gene and an AVR gene. The idea being that when the pathogen contacted the cell, it would activate this cassette, and you'd do HR, and you'd get resistance. Makes sense, right? So apparently, that's what was licensed, and this is the result of that experiment. Everything died. There was a little diversity, I have to say, but there was absolutely no effect of the transgenes. So we did see some diversity. When you put potatoes through a callus, and you release a lot of somoclonal variation, and we did see that. We saw nothing that we could attribute to the transgenes, which I thought again was really interesting. So the question I've asked is, why can we not see in the field things that other people can see in the lab? And I think the answer is this, that the sporangia from a plate culture are different from the sporangia that come from an infected leaflet. The reason I think that is that we had previously seen some suggestions. The first suggestion came with work done by Eduardo Mizubuti. And Eduardo's interest was in the sensitivity of sporangia to solar radiation. And Eduardo had a tough summer. He was getting sporangia from a plate. He'd expose them to sunlight, and absolutely everything died. They didn't survive at all. One time when he didn't have a culture from the plate ready, he took sporangia from a leaflet, exposed them to solar radiation, and lo and behold, some of them survived. So all the rest of his experiments then were done with sporangia that came from a leaflet. So the sporangia from a culture plate are very sensitive to solar radiation. The sporangia from a lesion are much less sensitive to solar radiation. So that's one difference. Another came from work we collaborated with Dan Klessig at BTI. Dan was interested in arachidonic acid stimulating resistance in potatoes. And we gave him an isolate of Phytophthora infestans. And these are small images, but you can see that if there was no arachidonic acid applied to the plant, there was a good sized lesion. If arachidonic acid was applied to the plant, then there was a really tiny lesion. Great. Patty Manasalvo published that. The problem came a couple years later when we tried to repeat some of that work. We got the following result. And we got the result because we took our sporangia from a leaflet, and this is what we have obtained. There was absolutely no effect of the arachidonic acid if we took this sporangia from the plate. We did the experiment properly. We had this control, and we got that result, that if we took the sporangia from a plate, then we saw the effect of arachidonic acid. If we took the sporangia from a leaflet, we did not see the effect of arachidonic acid. So that caused us to begin to look at the differences between sporangia that are produced on a plate versus sporangia that are produced on a leaflet. And what I'm going to describe next is some of Sean Patif's master's thesis work. So the first assay that we did was to look at pathogenicity of sporangia that come from a plate versus sporangia that come from a leaflet. And he did the work, first of all, in field plots using two different isolates. For isolate 1, he found if the sporangia came from a plate, he got 12% disease in the field. For isolate 1, if the sporangia came from a leaflet, he got 35%. Now, it turned out the isolate 1 had only 2 passages in pure culture. If he looked at isolate 2, which had been on a plate for at least 9 passages, he found 3% disease versus 52% disease. So there's quite a bit more disease resulting from the same number of sporangia, depending on where the spores come from. If there's little disease if they come from a plate, lots more disease if they come from a leaflet. Well, Sean repeated this experiment in a moist chamber, and he found somewhat similar results for isolate 1, 27% for sporangia from a plate, 80% disease for sporangia from a leaflet. For isolate 2 that had 9 passages, he found an even greater difference, 1% if the sporangia came from a plate, and 87% if the sporangia came from a leaflet. So clearly, there are differences in the aggressiveness of sporangia coming from plates or leaflet. It's interesting to me to note that for inoculations that are done in the literature, the concentration of sporangia in a medium can have a 3 orders of magnitude difference. Sometimes there are hundreds of sporangia per mil, particularly from our lab. And sometimes there are 100,000, 200,000, 300,000, 400,000, 500,000 sporangia per mil in inoculation. And my thought was, you know, they probably use 100,000 sporangia if they need them. And they would use fewer sporangia if they didn't need the huge numbers. That's never said in the paper, but that's a conclusion that one can draw. And then Sean also did some gene expression studies. He took sporangia from plates, sporangia from leaflets, and he did applied those to RNA-seq. And there's a little bit about the genome of Phytophthora infestans. There are more than 240 megabases in the genome. 75% of the genome is repetitive DNA. There are more than 17,000 genes. And in this analysis, we found more than 15,000 genes with a reads per kilobase of exon model per million base mapped reads. So those are the ones that we can detect reliably. How am I doing, Sanchan? Is that right? Okay. So our bioinformaticist is sitting right there. So we did detect a fair number of those genes. And what was just sort of blew me away were these data. Is that in the isolate of Phytophthora infestans, which had 9 passages on a plate, there were 4,791 significant differentially expressed genes. And that's comparing a plate sporangia to leaflet sporangia. For the isolate that had only 2 passages in pure culture, there were 2,155 significant differentially expressed genes, again, comparing plate to leaflet. Together, there were almost 5,400 significant DEGs, comparing plate sporangia to leaflet sporangia. There were, for these 2 isolates, there were 1,549 that were shared significant differentially expressed genes. Of those, 1,544 were expressed in the same direction, either all up or all down. So there are 5 that were not. That's a discrepancy rate of about 0.003%. It's interesting also that there were almost equal numbers of genes that were upregulated versus downregulated in these two different in this comparison. So both upregulation and downregulation. We looked at the RXLR effectors, of which there are more than 550 in the genome of P. infestans. And we detected 350 with an RPKM greater than 1. 170 of those were significant differentially expressed genes. That is, they were overrepresented compared to the whole genome to the rest of the genome as a whole. And they were both upregulated and downregulated. So there's tremendous diversity in the RXLR effectors. RXLR effectors are thought to be involved in pathogenicity in Phytophthora infestans. Some of them we know to be recognized by our genes. The vast majority, we don't know what they do. And I think also important is that there were no such effectors that were downregulated in one isolate and upregulated in the other. So they were always in the same direction for both isolates. So we had these pathogenicity differences, which I think were really solid. We had these many DEGs. And so we submitted a paper to MPMI as a technical advance, because I think it's important for people to know the phenotypic characteristics of the organism that they're working on. And then in August 19, 2017, we got this response that our manuscript has been denied publication. I find this part to be really discouraging. I hope the outcome of this specific submission will not discourage you for further submissions. Anybody else beside me have that reaction? So my initial reaction was this, that, you know, how could they be so stupid? But then I actually talked with Alan about it. So I went back and read the comments. There was one, that was nice. It was an interesting study. And the study does not explain the underlying mechanism. That's true. And then there was this. My principal methodological concern is that I would like to see more information on direct and indirect germination behavior of the sporangia. That's a reasonable request. Then there was another. The paper has a major technical flaw. The comparisons were made between sporangia from leaflets at 5 to 6 days post inoculation and from live P media at 10 to 12 days after inoculation. And so they had different ages of the sporangia. Actually, that's not a valid comment, but I had to address it. And then I found the analysis of differentially expressed genes to be a little shallow. Had to agree with that one too. So these are the objections. Not enough on indirect germination. The age of sporangia, there's insufficient analysis. So now back to the lab bench, and here I'm going to thank Kevin Myers, who did a lot of the next bits of work. Kevin, doing this was really fun, I have to tell you. Kevin and I had a wonderful time in the lab a year ago because we're getting all of these very interesting results. So the comment about indirect germination, that's an absolutely valid comment. So we looked at indirect germination. And lo and behold, that if you take sporangia from a plate, you get 3, 10, 8% indirect germination. If you take them from a leaf, you get 79, 40, 68, and 81. These are 4 different experiments. So there's a huge effect, just really a huge effect. So the plate sporangia germinate a lot less than the leaflet sporangia. And that could be a factor. Then we had to look at the age of the sporangia. The reviewer was correct. We used sporangia from a plate that was 12 days post-transfer, or 6 to 7 days from a leaflet, days post-inoculation. And so we looked at sporangia that came from 6 to 7 day old plates versus sporangia that came from 6 to 7 day old leaflets, after inoculation, that is. And these are the results we got. Exactly the same phenomenon. There was no effective age. But sporangia that came from leaflet germinate indirectly at very high rates. Sporangia that come from plates germinate indirectly at very low rates. So, I thought that sort of confirmed our initial title, that Sporangia, Phytophthora, infestans produced from a leaflet lesions differ from Sporangia produced from an agar medium. To me, we're just confirming what we had initially said. I want to show these data in a slightly different way. And this, again, I have to thank Kevin for. There's another way to look at indirect germination. And that is to look at the number of zoospores per sporangium. And Kevin did this. And again, the difference is absolutely huge. If sporangia come from a plate, the number of zoospores per sporangium is very, very low. If sporangia come from a leaflet, the number of zoospores per sporangium is about 100-fold greater. So there is a really big difference. Does that explain the difference in pathogenicity for these two types of sporangia? How can one address that?
Standardize the numbers based on the inoculum load
based on sporangia. Yeah, we could do that. What else?
Do you have data on direct germination?
I'm sorry?
Do you have data on direct germination?
Yes. Well, actually, we don't have data on direct germination, but we can prevent indirect germination. If we harvest, inoculate, and incubate sporangia at 21 degrees, that prevents indirect germination. Yes, sir, Adam?
Could you review for me the difference between direct and indirect germination?
Direct germination is when the sporangium, you're a bacteriologist. I mean, really. Direct germination is when the sporangium produces a germ tube. And that happens at temperatures above 20 degrees. Indirect germination is when the sporangium produces a zoospore, and that typically happens below 18 and is better at 15 or 10. So by changing the temperature, we could prevent indirect germination, which in fact we did. The results of 3 different experiments with an isolate with 2 to 4 passages on plates. And if you look at these experiments, you see that the number of zoospores per sporangium is really tiny, consistent with what we saw previously. When we saw the disease, we got the same results. Essentially, if the sporangium come from a plate, we get a certain amount of disease. If they come from a leaflet, we get 3, 4, or 5 times more disease. That was consistent with the second experiment, here about 3-fold greater disease. The third experiment, 10-fold greater disease. What Kevin did, and which I was really delighted with, is that he looked at the number of sporangia that were produced on the leaflets that we had inoculated. And what you see here is that, and we normalize that to the amount of disease present. So what you see here is that number of sporangia produced from a plate, about 1,000. Number of sporangia produced from a leaflet per unit disease was 15,000. Experiment 2, about 1,000 or almost 30,000. And experiment 3, actually got no sporangia, but about 16,000 from leaflets. So not only does that organism, is it less infective, but the lesion that it produces many fewer sporangia, probably less than 10%, as much. That was a difference I had not expected. I don't know if Kevin had expected to see it, but it was Steve.
Bill, have you ever tried to add some magic potion from leaves to the plates?
Let me get to that in just a bit. The question was, have we ever added a magic potion from leaves to the plate? No, but I'll get back to that. Okay, so what we found is indirect germination, there's much less from plate sporangia. If we prevent indirect germination, we get much less sporulation as well as less disease. There is no effect of the sporangia. And so our next task was to try to look at the amount of analysis that we didn't do. So we looked at the genes that were significantly differentially expressed that had flagellum somewhere in the name. There were 24 such genes detected. 14 were DEGs. 9 were detected in both. And these are those 9. And what you'll notice is that they are all in the same direction. That is, the expression of those genes in the sporangia from a plate is dramatically less than the expression of those genes in a sporangia from a leaflet. That makes sense, right? We then looked at elicitins. Now, elicitins, you will all know, are 10 kilodalton proteins that are produced by Phytophthora species. They do bind sterols. Their exact function in plants is really not known. And you know that if you inject them into leaflets, you get HR, right? We detected 41 with an RPKM greater than 1. 19 of these were DEGs, and there were 10 that were DEGs in both strains, GC here and V there. And so this illustrates the difference between plate and leaflet sporangia, the fold change. And what you notice is that 9 of these are dramatically more highly expressed in the sporangia from plates than they are from leaflets. 1 is dramatically less expressed in sporangia from plates than in leaflets. That's an interesting elicitin, I think. But these others are all associated with production in plates. We then looked at the highest expression, the top 40 genes. And again, the plate versus leaflet, the fold change. What I wanted to illustrate, these are the ones that are most highly expressed in plates. The ones with a green background, 3, 11, 22, and 27, represent sugar or solute transport, which is probably consistent with growing on a medium that contains a lot of sugar, which they did. There are 4 genes that are typical of stress response. The transferase nitrate reductase 2 heat shock proteins. There was one RXLR effector, which is interesting, and there are 2 elicitins, INT4, which is pretty famous, and one that has not yet been worked on. So that was a really interesting development. And then the next is that we looked at those with the lowest expression, the bottom 40, which means they were really highly expressed in leaflets. And interesting to this, there was 4 genes that looked like they were involved in signaling. There were 2 RXLR effectors that were very highly expressed. And there was another gene that was very highly expressed. It has to do with indirect germination. This has been previously published by Howard Juttleson's lab. So it seemed to me that we had done our work on indirect germination, no effect, or excuse me, large effects. Age of sporangia had nothing to do with it. And we had done some additional analysis. So I thought, surely, I'm going to submit this paper and it'll be just fine. We submitted it a little less than a year ago. And then on June 29th, I got this message back, which said, this version of your manuscript has been denied publication in Molecular Plant-Microbe Interactions. Again, I went crazy, right? Because we had done everything that the reviewers had said. So I'm going to show you next the comments from this second set. It were a different set of reviewers this time from the first time. So the method that our authors used to harvest the sporangia might have contributed to the difference. So what the reviewers was suggesting, when we rubbed the plate to get the sporangia off the plate, we got nutrients which would prevent indirect germination. And completely actually cited the wrong supporting article for this. So I did the experiment. I rubbed a plate without sporangia, put that on the leaflet and pulled that off, and it had no effect. So that is a bogus comment. And then the same reviewer said, usually isolates of P. infestans were maintained in the dark. It is true that some investigators keep isolates of P. infestans in the dark. That ain't what happens in nature. Yeah, actually, there's a diurnal phase in nature. But anyway, we did the experiment. We kept them in the dark or in an alternating photo period, and there was a Zippo effect. You know, it did not eliminate that effect. And this one I really love. It says, as we know, these sporangia from leaflet lesions were stressed by host immunity. You know, that's not what the RNA data said. The RNA-seq data said that the ones that were stressed came from the plates, which I thought was, so the dogma here is real. And so this is, I think, the inconvenient truth that Alan was describing, is that the sporangia from leaflets were actually not stressed. So I then wrote to John McDowell at Virginia Tech. So it happens that John McDowell is the editor-in-chief of MPMI. And so I'm asking, can you give me some advice on the manuscript that I submitted, et cetera, et cetera. Then I said, I know the manuscript is controversial, but despite the controversial nature, the data are really tight. And I explained to him about the bogus nature of the comments from this second set of reviewers. I did it nicely. We had some email discussion. Then he agreed that he would be a reviewer of another version. So I sent off the version on September 21. And finally, from the senior editor, the reviewers have recommended publication. So this paper is in press. This is figure 2 from that paper. And it really describes the differences. If sporangia come from a plate, indirect germination is lower, flagellar genes are lower, pathogenicity is down, elicitins are up, and stress response is up. If the sporangia come from a leaflet, indirect germination is up, flagellar genes are up, pathogenicity is up, elicitins are largely down, and the stress response is down. What is the real Phytophthora infestans? So I've modified my response to this question since I first asked it, and that is that, you know, both are probably real, but they are really different. Sporangia from a plate, I think they are aberrant. They're different. And sporangia from a leaflet are really natural. These are unstressed, those are stressed. I mean, this organism doesn't like to live on a plate. It really is, over evolutionary time, is a parasite pathogen. And it has a very significant phenotype associated with those differences. So what are the implications of this? The major one, I think, is that we need to investigate proteins. If we're interested in investigating proteins that are involved in pathogenicity, we have to look at those that are upregulated in plants and not that are upregulated in the plate, because there is such a huge difference. And we can avoid this description that I'll describe here. Again, 20 years ago in the plant cell, resistance of Nicosia and Benthamiana to Phytophthora infestans is mediated by the recognition of the elicitor protein INF1. What the authors had done was to take isolate or clonal lineage US1. They determined it was not pathogenic on Benthamiana. They silenced INF1, and then they got pathogenicity. What they didn't say is that this isolate had been on plates for about 20 years. It was dramatically different from anything that appeared in the field. At the same time, we were actually doing a little work on that ourselves in this very august journal, Plant Disease. And the paper's title was Host Pathogen Interactions Between P. infestans and the Solanaceous Host Calibrachoa hybridus, Patonia times hybrid, Nicosia and Benthamiana. All of these hosts were susceptible to P. infestans. They all produced INF1. So INF1 is certainly not the reason that, and actually Nicotiana benthamiana is not resistant to Phytophthora infestans. So this study is essentially irrelevant to anything that happens in the field. And I have to guarantee you that there is not a single sporangium from a plant that is involved in a natural infection or an epidemic. There is not one. One could look at targets such as these. These two RXLR effectors were very highly expressed in leaflet sporangia compared to plate sporangia. And there were many other genes that were really upregulated in leaflet sporangia compared to plate sporangia. So this is my final slide. There are at least two versions of this organism and maybe more. What's important is that you need to know which is which and identify that. I'm fascinated that P. infestans can sense its environment the way it does. And this sensing its environment really affects the phenotype, which is really, I think if you're interested in actually what might happen as a disease-causing organism, I think that's important to know. And it is also true that if you do work only with lab cultured strains, you could get misleading results. The question I have is, what about other hemibiotrophs? You know, is this phenomenon restricted to P. infestans? Or might it apply to other hemibiotrophs? I suspect it does not apply to necrotrophs. I think there's no way it could apply to obligate parasites. But to hemibiotrophs, I think there's a chance that others might have this same phenomenon. With that, I'll stop. And I want to thank you all for being here. I'm not going to thank any funding organizations because I didn't get any funding for this. And particularly, I want to mention Kevin, right here, who's done a lot. Hillary Mayton has been a fantastic support all these years. Sean Patif, it was his master's thesis that I showed you the work on. Margot Bechtel had published the work on Nicotiana benthamiana. And Paula Zuluaga was a co-collaborator in the proposals that were not funded through our funding sources. So with that, I'll stop, and if there are any questions, I'd be happy to attempt to answer them. Gary.
Well, I was curious, and maybe Sean's work addressed this, but you're contrasting growth of the organism on plates in the lab and on plants in the field. Did you ever look at plants in controlled environment growth chamber? And does that make any, is there something about the field in addition to being in plant, UV light, whatever other things might be going on in the field?
The leaflets can be either from the field or from the greenhouse. And that really doesn't matter. So the question was, is there something about plants growing in the field that might have an impact on these results? I actually think there could be, but the difference that I'm describing today is whether the plants were in the field or in the greenhouse didn't matter. Keith.
Bill, when you did the experiment incubating inoculating the plants at greater than 20 degrees, where you were inhibiting indirect germination, what was the germination phenotype at that temperature?
We did not look at that.
What were the sporangiophores from the plate even germinating at any frequency comparable to?
So the question that Keith asked was, when we did the experiment at 21 degrees, did we look at the phenotype of the sporangia as they were on the leaves? And we did not do that. Mark?
So when you did the passage to the plant of like the plate sporangia and the plant sporangia, and then you measured the amount that was on the plant at the end of that infection, do you know if this will keep going? Like, are those now at the leaflet stage, or are they still residually plate sporangia?
So the question is, do sporangia maintain their phenotype through repeated passages through a plant? I think, right? We don't know. That was a question one of the reviewers asked as well. And it's a great question, but I really wanted to get this paper published. So. Any others? No.
How do your finding of the two different types affect
the way in which plants are bred for resistance.
So the question was, how did our finding of these results affect the way plants are bred for resistance? I think the best answer to that is that those resistances that were, those sort of nuanced resistances that were detected in the lab, we could not detect in the field. And I think that's because the sporangia are so very different. So I think breeders they put their plants out in the field. So they don't suffer the problem that some would suffer if they never put a plant out in the field. So I think if you use leaflet sporangia, I think you can detect real differences. If you use only plate sporangia, I think you'll miss those nuanced differences, nuanced resistances. Alan?
So Bill, so in nature, sporangia are never produced except from lesions that began on living tissue. So I mean, you don't have any saprophytic phase that's comparable to plates in the sense of nature.
Yes.
So this is a completely aberrant program.
Would you like to say that again for the folks elsewhere?
Oh, yeah. So this represents what people observe or are working with plate-produced sporangia is essentially an aberrant sort of regulatory program that doesn't reflect nature. So, and one way to think about it too is,
yes.
yeah, okay, thank you. I knew you would, but another way to think about all of this is the shift from the biotrophic to the necrogenic phase, which appears to be occurring earlier with the plate-grown sporangia, right?
I think it does.
Yeah. Okay. Yeah. Okay.
Yes. Is there anything else?
Do you think that the spread from plate and the leaflets are genetically different or genetically identical?
So the question is, are the sporangia from the plate genetically different from the leaflet sporangia? I think they're exactly genetically identical, but their gene expression, their state is really different.
But I think that the experiment different because they were inoculated or grow at very different environments. So
Absolutely.
So that was environment triggered the different expression, not the genetically different.
Correct. So the environment, these sporangia respond to the environment, and what we see, the phenotype is a result of that response to the environment. Yes. How does one ask for questions from Geneva?
Just like that.
Are there questions from Geneva? I think not. Okay. Well, thank you very much. As you can tell, I'm kind of interested in this. I appreciate you coming.
This has been a production of Cornell University, on the web at cornell.edu.